This video continuation of structure of protein, already primary and secondary structure completed. This one is third level structure, which is tertiary structure of protein. What is tertiary structure? Tertiary structure is formed after the secondary structure has further folded and coiled by different type of interaction. So it is the three-dimensional folded structure. This one is the three-dimensional folded structure formed out of that peptide chain of the secondary structure. This structure is stabilized by hydrogen bond, hydrophobic, hydrophilic, van der Waals force and electrostatic bond, which means these are the bond which are found between hydrophobic and hydrophobic side chain, that is nonpolar side chain, and hydrophilic side chain, which are found in exterior. Now these interaction and bond are found between the amino acid side chain. Now this interaction and bond are found between amino acid side chain. So the amino acid side chain, they either attract, either repel, and leads to stable structure of tertiary. Now see the bonding. So this one is the non-polar. Okay, here hydrogen bond, here disulfide bond, here ionic bond. The non-polar group for interacting hydrophobic interaction. Non-polar interacting hydrophobic interaction. The OH group interacted with the O group to form hydrogen bond. Disulfide linkage bound, this one, disulfide linkage found between cysteine residues. These are the ionic bond. This one hydrogen bond, disulfide bond. This one is ionic bond. These are the three different type of bonding presented in tertiary structure. Now the secondary structure, alpha helix and beta bilated structure, they undergo folding. And form three-dimensional structure. Now all of us know that folding is responsible for function. Folding only responsible for function. Protein function only in tertiary structure except of insulin. So functions of protein dependent on tertiary structure. Now if the structure is disturbed, this structure is disturbed, protein are very susceptible to heat. So when the protein is heated, it's said to be disturbed to their safe. So it is called denaturation. We will give heat, the protein disturbed, so the safe also disturbed. That is called denaturation. Again, if you cool the protein, we cannot get safe back as well as structure also. So again we will do cooling, the safe and structure cannot come back. Just imagine egg, we boiled egg, we make omelette. This is because of denaturation of egg albumin protein unit. You cannot get back the egg from denatured omelette. Now how the structure of tertiary protein studied? We can study it by using X-ray and NMR studies, X-ray and nuclear magnetic resources studies. So this is all about that tertiary structure studying technique, X-ray and NMR. The example of tertiary protein, all enzymatic protein, all type of enzyme have an active site and the folding three-dimensional folding. All plasma protein likes antibodies. These antibodies, if heated, not bind with antigen, act on it. So myoglobin found in muscles. So summary of tertiary protein. This one is the three-dimensional structure formed by folding secondary structure. So it can be studied by using X-ray and NMR. Then examples are enzymatic protein, plasma protein, antibodies and myoglobin. Next, quaternary structure of protein. This one is fourth level of protein structure: primary, secondary, tertiary. They can be formed out of single polypeptide chain also. Apart from, they also form two or more polypeptide chain, but generally single polypeptide chain undergoes folding and coiling to form this three structure. But the quaternary structure, it forms from two or more than two polypeptide chain which will be linked together by non-covalent interaction. Example, electrostatic interaction, hydrogen bonding, van der Waals force and this type of bonding. Now the non-covalent bonding takes place in first seat. Now this polypeptide which are joined together, they are known as subunit. Still the time the subunit does not come together and not arranged properly, the protein will not be functional. Now, based on how many subunit present, they can be multiple subunit also, and this multiple subunit can either be associated with organic component or non-polar or non-protein part. This component organic or inorganic, that component inorganic in nature. That component is called prosthetic group. Multiple polypeptide chain are sometimes associated with an organic or inorganic, so which is known as prosthetic group. For example, in case of hemoglobin, the heme is the heme which bind iron. It is very important functioning of Hb. Then the quaternary structure, it can also called multi-protein complex or multimer. Heme ion containing component which form non-protein. What are the use of quaternary structure? Based on the type of subunit, that quaternary structure can be classified into based on the type of subunit protein can be classified into heteromeric. This one composed of different subunit. Then homomeric, composed of same monomer unit. Here different subunit A and B, but here same monomer A and A, like that only. What is the uses? Quaternary structure allows a protein to have multiple functions. For example, individual subunit may have different conformation and orientation which would enable them to perform function in a regular manner. Example, hemoglobin. One protein chain bind with oxygen and another chain bind with carbon dioxide. This is how one protein can serve to function. So here one chain bind with oxygen and another chain bind with carbon dioxide. This is how one protein can serve to function. Keep in mind these functions are done in a regular manner, just like a enzyme mechanism. Now the quaternary structure examples. The examples are very common. First one is hemoglobin in blood. Use, what is the use? For used for oxygen transport, it have 4 polypeptide chain. Then DNA polymerase enzyme used for DNA replication. Then lactate dehydrogenase enzyme which is used for glycolysis. Then collagen protein 3 polypeptide chain triple helix which are found in connective tissue. It consists of 3 polypeptide chain found in connective tissue. Then antibodies and different subunit of ion channels. Next, discuss about hemoglobin. It is a tetrameric protein. What is the use? Oxygen transport, quaternary structure of protein. It is a metalloprotein. Why? Because it containing iron. See the structure in clearly. They have two chain, two alpha chain, two beta chain. Alpha to beta 2 in it is associated with the prosthetic group which is known as heme. It has iron on it. Summary: quaternary structure is a compact structure. It formed by 2 or more polypeptide chain. Now this polypeptide chain called subunit. How many subunit present and what type of subunit present? They are called as multimeric, monomeric, heteromeric, homomeric. Also example hemoglobin, DNA polymerase, antibodies, ion channels. And HB is tetrameric blood protein which consists of 2 alpha 2 beta chain found in RBC of all vertebrates. So this is all about primary, secondary, tertiary and quaternary structure of proteins. Look at a statement form of primary structure of protein. Secondary structure of protein. N and N4th folding, but folding occur 3.6 amino acid. One coiled 3 and our next coil 0.6. Then beta related sheet structure. Tertiary structure of proteins. Hydrogen bonding, disulfide bond, ionic bond. Two functional group subunit is present. That means dimer. 3 subunit means trimer. 4 subunit means tetramer. Based on the protein subunit, it have different names. Example hemoglobin. Last one. Thanks for watching.